So the outline for the this part of the workshop, first I thought it would be useful to go through some basic theory, introduction to HDX, some basic theory, talk about the instrumentation and methodology used for these types of studies, and then the concept of differential HDX, which is mostly what we've been doing within the Hive, which is looking at either protein-protein interactions or protein-ligand interactions. We'll go through an example of HDX on the vitamin D receptor, which is a nuclear receptor, not related to the Hive. But here, the reason for this example is it shows a phenomenon known as EX1 kinetics, and Ji Zhang is going to go through this part, and he's going to show you, what that data looks like and how we can deal with interpretation of EX1 data. And then he'll continue with the cross-linking mass spec part using the RIG-I receptor as an example. We don't have any real live demos other than the EX. He's going to walk through some EX1 calculations on some Excel worksheets. The reason for this is the HDX software that we use requires it's PC based, and it requires a very specific configuration on a PC to actually work efficiently. Otherwise, it would have been completely useless for a demo. And there's, unlike the cryo situation where there's software that works with all cryo data, mass spec software typically works with vendor specific instruments. So what I would show you, what we're using, would only work for thermo instruments, and if you had waters instruments, it wouldn't work necessarily. So we just decided to eliminate that. So the general idea of HDX is that we want to use the exchange of protons within a protein kind of as a measure or a way to measure fluctuations in structure within a protein. So in other words, here's the protein under one conformation. The blue circles here represent amide hydrogens, and in the presence of deuterium, they're going to exchange. And what makes the technique useful is that not all amides are going to exchange at the same rate, and we're going to talk about why that's the case. By labeling, so really the protein conformational mobility influences the rate of amide hydrogen exchange with solvent deuterium. But the value or the reason behind this is that we've now isotopically labeled the protein, and then we can use mass spectrometry to measure this phenomenon. Now, there's lots of hydrogens in proteins. They can be binned into exchange regimes with the highly mobile acidic protons and the extremely slow to never exchangeable aliphatic or aromatic CH. So what we're really going to be talking about exclusively here is main chain backbone amide hydrogen exchange. So, and it'll become clear why, while we're labeling all these other protons, they're going to exchange out before we get to measure them by mass spec. So they're basically going to be invisible in the experiment. So as I mentioned, the idea behind this is we're going to isotopically label the protein and then do mass spec detection to report changes in the chemical environment of the amide hydrogen. So as it undergoes exchange fluctuations, it's going to report back to information about that region of the protein. It's also a solution-based technique. So we're going to take the protein in solution and expose it to D2O. The nice thing about that is we can fully automate it by using autosamplers, HPLCs coupled directly to mass specs, and using immobilized proteases to facilitate the localization of the exchange data to specific regions of the protein. And I'll go through that in a lot more detail in a few slides. We're also using high resolution mass spectrometers. This greatly facilitates the experiment in the fact that they have extremely high mass accuracy and high mass resolving power. So that allows us to do bigger protein complexes without as much spectral overlap, but also we can rely on the mass measurement once we've identified the sequence that belongs to that ion, because it's going to be within a few parts per million, so we can always find it within the chromatogram and be confident that it's that peptide. There are approaches. What I'm going to be talking about exclusively here is bottom-up HDX. Those familiar with proteomics, there's bottom-up proteomics and there's top-down proteomics. Bottom-up is where you use a protease to take a protein and fragment it into small peptides and then do your analysis that way. Top-down would be intact ion fragmenting using the mass spectrometer to cause the fragmentation. Top-down HDX is lagging significantly in that top-down has an efficiency problem in terms of getting complete sequence coverage across the protein, but it is improving, but it's still limited to a few labs and typically on proteins that are extremely well characterized. Middle down is where we would take peptides from a bottom-up experiment and then do ETD to sublocalize deuterium, and I'll just give one example. Software development is another key thing that enabled the analysis of large data sets, and I'll touch upon that in a bit. In terms of applications for HDX, Ingen had published an article in 2011 showing really where HDX has taken off over the years. From protein folding studies, which I'm not really going to talk. I give one example on IL-1 beta. Protein ligand interactions, that's predominantly what we're doing here within the Hive and what our lab is doing in general. In terms of the conformational characterization, a lot of this it has to do with comparing whether two proteins are exactly the same when they're produced. So the industry, the biopharmaceutical industry, is using HDX now quite a bit to show when they produce two batches of an antibody that their higher order structure is actually the same in solution, in the formulation solution. In fact, the last 3 postdocs from my lab were hired by Genentech, Amgen, and Pfizer, and they're all doing this for their job. In terms of our lab, I just threw this list up here just to give a general idea of the flexibility of the technique. It doesn't only work on one protein class. We've done a fair bit of work on GPCRs in collaboration with Ray Stevens and some other groups, a variety of enzymes, including with Dimitri recently on this Cas13D. And then some other studies, actually, Rigai I should have put up here. But so there's a lot of, it's a wide range of applications that could be, the technique can be used for. So that's sort of just why one should be interested. From a historical perspective, I like, Dimitri went over that, a lot of the aspects of cryo weren't exactly new. It was improvements over the time that caught people's attentions. People were using density gradient separation in the 50s. Lindstrom Lang really doing the first hydrogen deuterium exchange or hydrogen tritium exchange. Walter Englander as well, using gel filtration and liquid scintillation. And then in the 80s, Fred Richards decided to use digestion and HPLC for separation to kind of limit the exchange down to the peptide level. So that was the first concept there. And then Walter Englander was applying 2D NMR in the 80s. And the first use of mass spec came around the time with the revolution of protein mass spectrometry, with advent of electrospray ionization and MALDI top, MALDI with the Chait lab and David Smith's lab. But it was really, so there were, I had this publication graph, and I couldn't find that, but there were very few papers, maybe tens of papers up into the 90s. Once automation came into play in the mid-2000s, predominantly from the Woods lab, our lab, David Schreimer and John Engen, where the number of papers started to really take off. So this meant now that you can, one, run a lot more samples and generate higher quality data, reproducible data, etc. In the around the 2010s, it really started to take off into large, very large proteins. And this is when the instrument manufacturers had gone away from these gigantic high resolution mass spectrometers into the benchtop Orbitraps or benchtop QTOFs. So they didn't get cheaper, unfortunately. They kept the price tag, but they got smaller, and they got a lot more user friendly and higher resolution, higher sensitivity, etc. So this really was driving the next boost. So the papers have gone up dramatically since 2010. So just going into some general ideas of what are we talking about with HDX. So, you know, we are talking about isotopically labeling, you know, converting or exchanging hydrogen for deuterium as a way to isotopically label. Then we can do a mass measurement. If an amide is not protected from solvent exchange, this event is rapid. It's referred to as the intrinsic exchange rate. However, if an amide hydrogen is involved in a hydrogen bond, it's incapable of exchanging with solvent deuterium. And so the exchange reaction would be protected. Now, of course, proteins aren't static. So we know that there's an opening and closing of or breathing of the protein, whatever you want to call it, where this hydrogen bond is going to be broken temporarily. This is a slow event and a rare event. But when it happens, in the presence of deuterium, you get an exchange of that amide hydrogen with deuterium. So hydrogen bonding and solvent exclusion are the major factors, with hydrogen bonding being the most, the biggest factor of protecting. You do need solvent accessibility to have an exchange reaction. Without the solvent, you can't have the exchange. But you can have a fully solvent exposed alpha helix that is extremely protected to exchange due to the hydrogen bonding. So to put this into visual concept, here's a video showing you. So what we're going to see is an alpha helix here. And at the moment, this is water. So these are amide hydrogens involved in hydrogen bonds. Right now, you don't see any exchange. As they start to open, you'll see exchange. So the hydrogen for hydrogen will happen at a rate of opening of this helix or these hydrogen bonds. And then if you remove or dilute the sample or float away the H2O and put in D2O, what's then going to happen when those same bonds break? You're now going to get in exchange of deuterium, and you notice not all of them exchange. So some of them may have been broken for a longer period of time due to the strength of the hydrogen bond, etc. So that's basically what's going on. That's an isolated helix, but in the context of a full protein now, which is going to be much, ooh, I knew I was going to do that at some point. In the context of a full protein, it's going to be slightly slower because it's going to be more stable than, say, a peptide of helical structure. So as this moves around, we see now that the same phenomenon is happening, but it's now going to be happening at a slower rate. And then if we look at this same helix now in the content, say, of a homodimer, where those helices are interacting, the overall complex is much more stable. So the opening and closing of these hydrogen bonds is much slower. So now the rate of exchange is going to be that much slower. So that's the general concept. What we're going to be talking about when we go through how we do the experiment, we're going to take these proteins that are labeled under native conditions. We're going to fragment the protein and then analyze it by mass spec. So some of the factors that influence, besides hydrogen bonding and solvent accessibility, that greatly impact the rate of exchange is pH. This is an acid-base catalyzed reaction with a minimum around 2.7. So for every pH unit increase, there's an order of magnitude increase in the rate. So typically for most experiments, we're doing the labeling in the range of 7 to 8. So it'll be your protein. If you're doing a complex interaction and it's typically done at 7.5, the exchange experiment will be done also at pH 7.5. However, if we look at the rate of exchange at pH 7.5 and 25 degrees C, the rate is very high, and the half-life of those deuterium, so if you took it away from a deuterium environment, is extremely short. So we wouldn't be able to do any kind of mass analysis. So what we need to do for the experiment is reduce the pH to the minimum, to basically reduce by almost 5 orders of magnitude the exchange rate. And then what this does for us is the half-life now gets into the tens of minutes, but that again, it would be at room temperature. If we go to 0 degrees or close to 0 degrees, we get a few orders of magnitude more. Now the half-life of that deuterium is around almost 2 hours. So that's going to be critical for us to be able to do the mass spec experiment and not have lost all the deuterium in the. So we're going to be doing our labeling under native conditions, or whatever conditions your protein's functional at. If it's pH 6, we would do it at 6. But all the subsequent steps post labeling are going to be by adding a buffer that lowers the pH and keeping everything cold and doing the subsequent steps. So if we look at the overall process, again, if this amide hydrogen is involved in a hydrogen bond, there has to be an opening event to allow it to be exchange competent. There'll be a chemical exchange with deuterium. But the counter here is the closing rate. So the rate of exchange is the equation is shown here. For native state kinetics, what's happening is the closing rate is much faster than the chemical exchange. So what you would see here is a slow buildup of deuterium over time. And again, this is considered native state HDX. If for whatever reason the protein opens and doesn't close in a rapid fashion, and the closing rate is much faster than the chemical exchange is much faster than the closing exchange, this would be EX1 kinetics, and I'll just show you what this looks like. So what you get for that in this EX1 here is a bimodal distribution, and what you're seeing is molecules that have yet to exchange and molecules that have fully exchanged. So over time, you're going to get to where it's essentially fully exchanged. But we're going to say more about this later, and G is going to go through an example and what this is telling us in terms of the conformational flexibility of the protein. EX2 kinetics will look like this, where you see a gradual buildup in some cases, or it can be a rapid exchange if there's little protection, but there's a lack of a bimodal distribution. It's not necessarily Gaussian, and in fact, there's information in the fine infrastructure of this highly resolved isotopic envelope, but that's beyond discussion for today. So if we want to do calculations of protection factors, how protected is a region, and what does that mean in terms of the protein flexibility and dynamics, we'll go through this concept here. the intrinsic exchange rate is basically if you had this amide in an unstructured peptide and it wasn't protected by any hydrogen bonds, it has a certain rate of exchange associated with it. And if you actually do all combinations of dipeptides, you'll see that it's actually a protection factor, and it's about an order of magnitude between alanine to, say, tryptophan, lysine, for instance. So it's both steric and electronic effects that influence that exchange rate. Oh, sorry, that was this example here. So the rate of exchange for each dipeptide in combination was determined by NMR, and so that gives us the K intrinsic, the intrinsic second order rate exchange constant for an unprotected or amide. So what this would really mean is if the region you're looking at in a protein matches that of the intrinsic exchange rate, you could say it's disordered. It's random coil. It's not in the structure. There's no protection to exchange. And you can calculate the intrinsic exchange rate from the fact that Walter Englander recorded the intrinsic rate for all dipeptide combinations. So you just basically plug it into an equation. It's the protein sequence, the pH, the temperature, and then you just minus the two N-terminal residues, because now once you cleave the peptide, the N-terminal amide is now an amine, so it's no longer protected, rapidly exchanged. And he had shown by NMR that the next amide is influenced dramatically by electronic effects of the amide. So often that's fully back exchanged. So, and of course it would be minus prolines, which don't have the amide hydrogen. So going back to this, where we have alanine going to, say, like tryptophan, that's some protection factor. It's approximately 10 to the 100 or so. If we then consider this amide within a folded protein, what we notice is that the protection factor could be high as much as 10 to the 8th. And that's what actually makes the technique useful, because this is a dramatic shift to the right in the rate, which allows this to be easily measured, first of all, and also reads back on regions of the protein that are highly ordered. So if we look at that in terms of actual, even though these are hypothetical, but the way the data would look like from what's being measured, this would be time points of exposure to deuterium. This would be deuterium level determined by mass spectrometry. So this would be intrinsic rate of the alanine, and this would be, say, tryptophan. So there's that protection factor. But then when you look for that amide, sorry, in the folded protein, that's where you can see this huge shift. And then if you then add a binding partner, there could be a further protection to exchange or protection factor 3. So that's sort of the concept here. This is the intrinsic rate for your peptide, and you measure it in a protein and it overlaps perfectly, it's not protected. If it's shifted to the right, it has some degree of protection. It has some secondary structure usually, and or is protected by hydrogen bond networks. So in terms of what can we glean from this information? So again, if there's no protection, no amide, no hydrogen bond, so we can calculate delta G of chemical exchange, but we measure this here, the actual observed exchange rate. So the chemical exchange, again, is related to the intrinsic exchange rate. So you can calculate this guy with this equation. We measure this, so you can calculate K delta G EX. And so if there is a hydrogen bond, you can then calculate the overall delta G for opening that region of the protein using the following equation, and that KEX over K intrinsic is the protection factor for that region. Now, what should the HDX data look like? We were showing these ideal sigmoidal curves, but it's actually a mixture of all the exchange rates of all the amides within the peptide. So again, I don't know how much I emphasize that, we're at the peptide level, not a single amide. So if it was ideal and there was this sort of mixture of these exchange rates, you would have this kind of curve. So there's information in the actual shape of the curve. So if you have enough sampling over the time window, you can get to binning how many really fast exchanging, medium exchanging, or slow exchanging amides are within a particular peptide. So again, if you look at ideal data, here's an intrinsic exchange rate curve, and then in a folded protein, it's shifted to the right, it's protected. That's easily determined, even with the time window shown here, with 10 seconds really being a practical limit for what we can measure in the laboratory time frame. So here we have a full curve, so you can really have accurate measurement of the protection factor. But what about a peptide that's not protected to exchange? Here's its intrinsic curve, but our earliest measurement is here, so we only have partial data over the time window. So in fact, there could be protection in this region here, and we're just not going to see it. This will happen, say, in intrinsically disordered regions of a protein that are undergoing rapid changes. You know, they're stabilization, but they are in equilibrium between unfolded and folded, and it's a rapid effect. So there are ways to deal with this, and it's often referred to as time window expansion, and that's taking advantage of the pH effect. So if you generate data on a protein at pH 7.5, this is the experimental time window that you're in, but with your analytical platform under the same time window or time points, if you're at pH 6, you've slowed down the exchange rate, so you effectively start collecting data in the earlier time window. So there's caveats to this. The pH change has no effect on the protein stability. And one way to determine that is that the curve should overlap between what you've generated at pH 7.5 and pH 6. If they are offset by quite a bit, that means there's been an effect of the pH change. And there's other biophysical measurements you can do to show that pH change hasn't affected it. But so we've done this in cases where we're looking at intrinsic disordered regions of proteins that are important for interacting with either DNA, RNA, or other transcription factors. So that was all cartoons and ideal data. Here's some real data. Again, here's the experimental time window we're talking about. Here's the intrinsic exchange curve. So in this particular example, there was a peptide, a protein in the absence of any ligand. This particular peptide, that's its exchange behavior, so it's protected. There's hydrogen bonds being formed. There's some structure in there. And then we added two different ligands. One of the ligands didn't affect that region at all. It overlays perfectly, whereas the second ligand, you see protection or a shift to the right in the exchange kinetics. Yes. Yes, that's calculated based on Walter's, the NMR data. You put in the sequence, pH, temperature. No, primary sequence. Yeah, so it's assuming random coil. So if that sequence of had it had no structure would be its intrinsic exchange rate. So each dipeptide is slightly different, and the pH at which you're doing the experiment and the temperature. Minus prolines and minus the 2N-terminal. So again, if this particular peptide within this protein had no structure, was not protected, it should overlap, and it would be probably like from here to here. It would just be flat. So, whoops, that's the next slide. Sorry. Oh, so then if you consider this in the presence of this other ligand, there was protection. So you know that would be you could calculate the protection factor difference between, so one, you can calculate the protection factor for that sequence of the protein within the folded protein, but then you can also calculate the protection factor between the ligand bound. And often what we're always doing is importing this data and overlaying it onto a PDB structure, whether it's a model or an actual structure or a homolog or whatever. So that was just the basic introduction theory. There might be more things that come out as we go through this, but the next thing is instrumentation and methodology. But before we go through that, just want to really talk about the workflow. So you give us a protein. The first thing we want to do is digestion optimization. So this is where you have your protein, 2 proteins, and you want to look at the interaction. We need to generate the peptide set that we're going to use in the experiment to measure their exchange characteristics. So we take that protein and we use a series of quench conditions. That's, remember, we're labeling at 7.5, and then we want to bring it down to 2.5 to prevent further in exchange or out exchange. So we have a 5 cocktail quench, different quenches that we use, various denaturants, with the idea that we not only want to lower the pH, we also want to get the protein ready for proteolysis. So we run that protein through that. The sequence coverage is determined by MSMS, which I'm going to just go through for those who may not be aware of any of the mass spectrometry part in a second. We use a long gradient here to just basically get as much MSMS experiments as possible. It is done under HDX conditions, so low pH and low temperature. So the fact that we're doing this on low pH, you guys are familiar with using trypsin, lyc, whatever, when you do your proteomics experiments, they're not going to work here at all. So we're using pepsin and some other acid stable proteases, which I'll mention another one in a second. And because of the fact that we need to do this fast so we don't lose deuterium, the digestion time is less than a minute as the protein flows over. So that's why I said we're doing a poor proteomic experiment. So we have a good chunk of protein that's undigested in a lot of cases, and that hits sensitivity to some degree. You can extend the digestion time point at the expense of losing deuterium. So everything we're doing, you can modify at the expense of losing deuterium, and if you have a big change in protection, then you can get away with it. If you're looking for subtle changes, as you lose deuterium, it gets into the baseline and you don't have any signal. The MSMS data is then put up against MASCOT. These are like MASCOT and sequest or database correlation algorithms used by proteomics. It correlates the peptide ion or the ions that are being measured using their fragmentation data to a protein sequence. And then this data becomes the peptide set for all subsequent experiments. The next thing we then need to do, so we may have 100% sequence coverage here, is we generate what we call our T0 map. This is would be the first time point in your HDX experiment, which is basically no deuterium. And it's under the ideal HDX conditions, which are low pH, low temperature, but also a very short HPLC gradient. So we're doing 6-minute separation. You know, your proteomics score is doing 2 to 3 or 4 hour separations to get maximal proteome coverage. Here we have to do really short, otherwise, again, we won't have much deuterium at the end. And we use this to confirm that we maintain a high sequence coverage with using the peptide set. Then we run our ion exchange experiments. These are the multiple time points in triplicate, and then the data processing. And this is where everything comes crashing to a halt. So you can do all this stuff really fast. Here requires, now the software is advanced significantly in terms of doing the peak detection within each run and replicate using that peptide set. I wrote very time consuming. That was for the next one, actually. The manual inspection is what's time consuming. You still need to go in and make sure that the software had used the proper retention time to get the maximum signal for that peptide. And the centroid window often, in some cases, will have to be adjusted to avoid overlap, spectral overlap, because if there's very close mass peptide in the centroid window, it's going to include that in the centroid calculation. There'll be an error in that measurement. And then the subsequent %D calculation statistical analysis is essentially extremely rapid. And then the data rendering is really just up to what you want to actually accomplish in the experiment. So, yes?
So maybe I missed this, but what governs your standard efficiency? Proteins that, or your coverage.
Yeah. Right. So couple things. One is, can we find a quench condition that under this relatively short exposure to the pepsin column or the panthecin column is sufficiently denatured that we get lots of digestion of the target protein? So in other words, we have to generate those proteolytic fragments to get the sequence coverage. So if we get poor fragmentation, we have poor sequence coverage.
Can you use combination of proteases?
Yeah, we do. We have tandem columns of pepsin and the panthecin too. And I'll show you the main difference between those two is pepsin will not cleave at all at basic amino acids, whereas the panthecin will cleave those. And as it turns out, with RNA binding proteins or enzymes, active sites of enzymes usually have high number of arginines and lysines. What happens with those peptides is, one, they might be big, they have a lot of charges, so they're high charge state, they fragment poorly, and then when you do the database correlation, you get a very poor score, so they're eliminated. Now, our software will go back, because we have such high mass accuracy, we can rely on the MS1 data alone, the mass measurement, but that comes with a risk, right? We don't necessarily, we could go through and manually inspect and see, well, maybe that MS vector is indicative of that sequence. But what we found is that if we use the panthecin too, we eliminate a lot of the potential artifacts that you may have. And the other problem with really high charge state peptides, even if you identified them, when you do the ion exchange experiment, if you have like a +5 ion, it starts to get spectral overlap, usually with peptides in that region. Sorry. So just to rehash the workflow, we ID the peptides using MS, and then we generate our peptide set and we're ready to do the experiment. We're going to expose the protein to multiple time points of deuterium exposure. We're going to quench that reaction to slow exchange, what we call slow exchange conditions, low pH. We add denaturant to facilitate the proteolysis. As those peptides elute off the column, they're electrosprayed, and this is just an example of a cartoon of what the isotopic envelope may look like. The data processes it and it generates the percent deuterium over time, but it could be percent deuterium, it could be delta mass, it could be anything you want it to be, really. So we typically use delta mass or number of deuterons or percent deuteration as the measurements, or as the values on the y-axis. So now going through the instrumentation, I mentioned this already, the MS is done using, the amino acid sequence identification is done by MS. The ion exchange experiment is just basically MS, just mass measurement once we've identified or correlated the peptide ions to the sequence. But we're doing LC-MS, so when you couple HPLC to a mass spectrometer, this is a hybrid technique. It amplifies the problem of each one of the techniques. So anyone doing a lot of LC-MS knows this. Any part of that system can go bad at any point in time. You can clog a column, the ion source gets dirty and whatever. So we do a lot of maintenance and a lot of QCs between runs, but it's just something to keep in mind. So not that we like to give excuses, but we often have to give excuses as to why we need either more sample to rerun it because there was a failure. I'm sure everybody here knows a lot about HPLC, so I just threw this up here for the biophysics class that I teach. You know, we're using capillary LC systems, relatively low flow, not nano flow, because that wouldn't be robust enough for this experiment, particularly since we're doing very large protein complexes often in complex buffers. Our quench buffers are quite harsh, and we're running multiple time points on a lot of replicates. So we need the system to be robust. So we're not doing, you know, what the proteomics core would be doing in terms of nanospray. The mass spec instrumentation, we have several Orbitrap instruments. I'm not going to go through this. There's a nice video here. I guess everyone has access to the slides. Is that right, Art? Yeah, or at least. So if you click on this, Therma has a nice video of how the instrument will do mass measurement, will do MSMS. But basically, the mass spec is composed of the ion source. This is where the HPLC is going to be coupled. Electrospray happens here. Ions get brought into the instrument, focused. Neutrals are removed. And then in the first region here, you can do mass selection for an MSMS experiment, fragment it, and then push them into the mass analyzer. For MS1, you're just going to push the ions into the mass analyzer. So that's the general idea, but to make it even more simple looking, MS1 data is generated by, you know, you take your protein and you ionize it, or peptides, into ions. Oh, sorry, protein digestion, you get the peptides, you ionize them, and then you put them into the mass spectrometer and you just measure them. So what this would look like at any one time slice from the LC is you have a lot of ions over various mass to charge ratios, and what you're going to do is do co-adding over a certain number of microscans, and then generate a mass spectrum that looks something like this. So this is just an example of an electrospray peptide where the predominant ion is a +2, so there's 2 charges, and then there's a +3 ion. So that's MS1 data. So in the ion exchange experiments, we're just going to be measuring changes in the centroid of these peaks. We're no longer going to be doing MSMS. But to do the identification of that ion to its sequence, we need to do the MSMS experiment. So there, in the tandem mass spectrometer, what happens is at least one component of the mass spectrometer is doing an isolation of the ion. There's a component of the mass spectrometer that does the ion activation to fragment it into pieces, basically, using either high collision dissociation or collision-induced dissociation. And then the fragments are separated and detected to give you this sort of, it used to be parent and daughter. I think it's now precursor and product, to not offend anybody. But basically, what this looks like now is in your mass slice, your time slice, where that peptide's coming off from the HPLC, you're going to select that guy. You're going to collisionally activate it and record its fragmentation. Now, these Orbitrap instruments do this by selecting, in the older versions, top 5, second generation top 10. Now we're doing top 50 on the Lumos. So for every single run, it could do 50 MSMS experiments. So this generates a lot of data, but so just to give you, again, that's a cartoon, but here's a real time slice of an MS1 spectrum. So there's a lot of things going on in here. If we took the top 50 here, it's going to select all these guys. It's going to isolate them to get rid of everything else. So now it's really just this ion, and it all depends on your isolation window, but it's fairly homogeneous. It'll collisionally activate the ion, and it fragments into these pieces. And then, if you remember from your biochemistry course, the residual mass of every amino acid, you can just use a calculator and walk through this and say, oh yeah, this is a partial sequence. No one likes to do that anymore. I did that for my thesis in Don Hunt's lab, but that alongside John Yates, he hadn't developed sequest yet. So, and there's rules for fragmentation, and it often depends on what type of mode you're using. I'm not going to go into any detail here, but just showing you that ETD, ECD, and collision activation do give you different fragments. But So what we're now doing, we're all spoiled. We take our protein, we digest it, we fractionate it, we generate MS1 data at a very high duty rate, 10 hertz here, we're gonna get 10 MS vector per peak fragmentation. We then push that over to a computer on either Sequest or Mascot, and then what comes out is a sequence coverage map for a protein. Now, in a proteomics lab, if you're doing you ran a HEK cell lysate, you're putting it up against the non-redundant human protein database. For our experiments, these searches are really quick because we're only putting the proteins in that are in theory in the sample. There's no, now we do a decoy database, which is either, it's just a database we have, which is reverse sequence of a bunch of proteins to get the false positive, false discovery rate. But these searches are super fast compared to anything you're gonna get from the proteomics lab because the number of proteins is small. And this is just showing you for the same protein under two different quench conditions, where the quench condition is written in here somewhere, but I can't see it because of my poor eyesight. But under one case, you know, we only had 42% sequence coverage, but in this other quench condition, we had 100%. So that quench condition does have a big effect on can have a big effect on the sequence coverage. You can display that coverage map by oh, another key thing is this is just showing you the sequence coverage, but what's actually happening is there's lots of peptides. You'll see regions of high density peptides throughout the protein sequence. So in this view, you get the idea of how much overlapping peptides there are. And this is important for, I'll show in the data analysis part, reducing this down to single amino acid exchange rates. There's multiple ways to do that. And what I'm showing here is two different proteases we use, pepsin and nepanthensin 2. What you see, the first thing that's obvious is they, in general, look the same. But when you go and blow up a region, here the red arrows are the pepsin cleavage sites. So nepanthensin kind of cleaves at those, but you're getting much more cleavages around basic amino acids. And this has helped us in several proteins. Again, where you know, as fate would have it, if this is your protein of interest, you're really interested in this region and that peptide's missing. So now it's like the sock in the dryer. Every one of these peptides has got to be there somewhere, right? So there could be a couple of reasons. One is I think I don't really show the valving, but there's a series of valves here. So small peptides may be just lost and never brought into the mass spec. Those that are very early may be just suppressed by all the urea and everything that's in the quench buffer. And then you may have some really hydrophobic peptides that just never come off. So there's those, but then there's these highly charged peptides that just score poorly in the process. But this generates our peptide set. So we have a retention time information, we have a charge state, we have the, it's not shown here, the exact mass measurement within 3 parts per million, and the sequence that was associated with it, and its score in Mascot. So we have a score of 20 threshold, was really conservative, but these are like super high scores. So, you know, there's unambiguous assignment of those peptides to those ions. So we're going to use that peptide set now in everything else we do. So again, remember, we're going to be labeling over various time points. We're going to quench it to slow the reaction down, generate peptides over the protease column, mass measure those. As I mentioned, it's a poor proteomics experiment because we're doing really short chromatography. But because these instruments have gotten so good in terms of their resolution, we really haven't seen much in the way of sequence coverage suffering there. But we've, to be able to do a bunch of samples, we rely on automation. So we have a sample handling robot that will pick up the protein solution, bring up the D2O addition after the incubation, and it will pull up the quench, mix that, and then inject it into a series of valves that the first one, using an isocratic pump, pushes it over a mobilized protease column. It gets trapped on a C18 cartridge. Then the valve switches and the gradient pump elutes that off into the mass spec. This is what the system looks like, because again, we want to keep in the earlier iterations of this, we just had this portion under temperature control, but you really want is the labeling and quenching also on low temperature. So we just decided to throw the whole thing into a -4 deli fridge. Leap freaked out at first because they couldn't guarantee the system would work at 4 degrees, but they've been going now for 5 or 6, 7 years now in our lab. Very reproducible. So the only modification we had to make to these deli fridges, drill this tiny little hole to allow the from the HPLC column over to the mass spec tubing to go through there. Now, some labs will be really fanatics and put cooling tape on that. It's really short in our case, and we really see almost no difference in deuterium retention whether we cool that or not. So now let's go to the mass analysis. So that's a part in here that I didn't really talk about. Not the instrument, but what it actually looks like. So remember our doubly charged peptide spectrum. What I didn't show you is when you blow that up, you see that you have isotopic resolution on these high resolution mass spectrometers. What this means is that here's your monoisotopic mass for this peptide. So this is your, carbon 12, nitrogen, hydrogen, oxygen, sulfur, etc. But for every, there's a percent carbon 13 for depending on the number of carbons, and then you get this peak here plus the other isotopes. So every peptide will, sorry, this is a deuterated peptide. Wouldn't exactly look like this. It'll be, oh, actually I had a cartoon of that a minute ago. Here, it'll look like this. For most peptides, the bigger they get, the more it skews this way. But this is a deuterated peptide, so this is what it looks like. The software knows the M0, so it knows the mass, the monoisotopic mass. It knows, based on the number of amides, what 100% should look like. So it sets bars for MZ start and MZN around that to do the centroid. So it's a mass weighted average centroiding. And this gives you a value that when you plug it into this equation here, will tell you how much deuterium is in there. So it's M versus M0. This is the measure. This is 100% minus M0 times 100. You can also do a theoretical modeling, and the software can do this, but it's really slow. Basically walks through all 0 to 100, and then does a chi-square fitting. And in this case, 43% is the best fit. The centroid approach is much faster, and the results are extremely similar between those two. So I don't want to, I'm almost at an hour, so I need to give G some time, but I'm almost done with my part. So just as an example for IL-1 beta, so this is the digestion optimization, 2 different quench conditions. You can see one gives you a higher density and more sequence coverage, so you use that guy. You do your multiple time points. You use, you determine the deuteration level. You can color code this. So we often refer to these as HDX heat maps. These are the 2 amino acids that we have to drop off. If we had overlapping peptides, you see this continuum here, and you can see this mixture of colors. So there's the N-terminus, which is basically not protected. Slightly in from the N-terminus is highly protected. Then around 20 to 30, it's moderately protected, and then again, not protected between 30 and 40, and so on and so forth. So again, for this one particular peptide, residue 152 to 154, that's the C-terminus. Here's the measure, and this is the intrinsic exchange rate. So there's no protection to exchange, so you could say it's disordered random coil. But if you look at this peptide here, 144, that's this guy right here, you can see that there's a significant protection to exchange from the intrinsic exchange rate. So when you plug in those calculations that I showed you before for protection factors, you can overlay them on the structure. In this particular experiment, it was looking at the basically melting of IL-1 beta at different pHs. And I did mention, though, this time window expansion issue, which I showed that slide earlier. So you can get additional information by doing this at the different pHs. So now I'm going to just go quickly into differential HDX. So the workflow looks identical, except now we're doing sample A versus sample B. and we're looking for regions where there's changes, we can overlay that onto a heat map. So now this is just not the intrinsic exchange or the exchange behavior of the APO. This is the regions within the protein that are differentially exchanged, and then you can overlay that on the three-dimensional structure. So the typical experiment is about 7 hours because it's 42 injections. They're randomized. There are 3 independent biological replicates and 7 time points each. We can modify this depending on the questions being asked. But this is the slide that's most important to potential collaborators. We send this out to you if you're interested in doing an experiment, because this walks through the actual amounts of protein that we need, and the concentration that's ideal, and things we don't want in the sample. Now, we do GPCR work. We are using detergents, but there's certain detergents that are not tolerable. So basically, anything your proteomics core hates, we're going to hate because it's going to interfere with the electrospray process. So then you can ask a question, well, how reproducible is this data? So we had internal data because we had a big project, and G is going to walk through this example on vitamin D receptor, where we had run several, maybe 300 to 400 ligand interactions. So we had a lot of APO runs. So if we just looked at the 30-second on exchange or 127 replicates, you can see that majority of the data was within 10% of the mean. But then we thought to be more useful would be to compare the same protein, a lot of the same protein sent to 2 labs, one in Spain and one in South Florida. This is the overall HDX map across the protein sequence, and this is looking at one particular peptide, which is, I believe, 15 experiments data here, and you can see that it's highly reproducible, and this was published in Analytical Chemistry. So independent of lab, the instrument platforms are slightly different. If you're doing the experiment correct, you should get very similar results. Now, it would vary from lot to lot if there was some influence on the protein. This was done with the same sample lot, but this is really the basis for the companies using this to look at antibody production and bioprocessing. So data analysis was huge for us. I'm not going to walk through any of the gory details, but it's been published. We needed a way to handle a lot of data, so a lot of runs, a lot of replicates, and calculating all this data and reducing it. So this is just sort of a general idea of what's going on. It'll take all the APO versus ligand data to give what we call a perturbation view, and then this would be a perturbation map. We then use the overlapping data here to generate a single amino acid level consolidation. There's lots of caveats within this, but the reason for doing this is we need to overlay, if we want to overlay this on a PyMol, we have to have one value per residue. We can't overlay multiple peptides onto the PyMol. So, but I took the slide out here. We've done a Bayesian approach to calculate single amide resolution, and they're almost the same. That takes forever. This is basically ones and zeros, so it's extremely fast. Takes no computational time at all. You can get actual single amide resolution data using ETD. This was the sort of middle down where you take the peptide and then sub-fragment it with ETD. The reason between having to do a different fragmentation versus CID is that CID imparts a lot of energy into the peptide, so you get scrambling of the deuterium, whereas ETD does not. So there's no scrambling of deuterium, and we had shown in one example here that you can actually localize within a peptide where the deuterium is being retained. The software also ports data right out to Prism, so you can do your statistical analysis. You can present data, say peptide APO versus ligand. If you have lots of ligands, you can do two-key analysis, etc. Across which ones are more similar than others. So what does the data on a real protein kind of look like? This is vitamin D receptor ligand binding domain interacting with its natural ligand, 125 vitamin D3. This is what we call the perturbation view. So this is APO minus the ligand data. The colored regions are where there's protection. Basically, all the regions that are highly protected correlate to regions that there's hydrogen bonds being formed between the ligand, with some exceptions that are related to stabilizing the homodimer in solution. This shows you how dramatic the protection can be. So this particular peptide within helix 3 of the protein has moderate exchange characteristics. When you add 125D3 over the entire time course, it basically doesn't exchange at all. So there's dramatic protection. You can overlay it onto the three-dimensional structure, so it all basically it correlates nicely with surrounding the ligand binding pocket. But then when you even just look at the APO kinetics in a video, what you see here is interesting. The protein, while the APO crystal structure shows a high degree of order, when you look at the APO protein in the absence of ligand, the exchange behavior, the bottom part, or the ligand binding pocket, is very dynamic, whereas the north end is highly stable. And over time, of course, this exchanges out more rapidly. So you get a nice idea of what the APO solution of the protein looks like. Now, if you have a protein and you don't have structure for the whole thing, what can you do with the data? I just showed this one example here. This is progesterone receptor. This is steroid receptor, where the ligand binding domain and the DNA binding domains are highly structured, so they've been crystallized. The hinge region that connects these two in the long N-terminal domain are resistant to crystallization due to the fact that they're intrinsically disordered. But you can actually do differential HDX exchange data and then just draw this out across the sequence. And in this particular experiment that was published here, we showed Tatabax binding protein interaction in the N-terminal domain PR. We were able to pick that up by HDX. So the VDR example, I'm just going to introduce it first, and then G is going to come up. Nuclear receptors are ligand modulated transcription factors. They're under complex control between ligands, interaction with co-regulators, PTMs, DNA binding, etc. So my lab focuses a lot on this. So the guys never run out of things to do because of all the different functional interactions. But in the case of VDR, what we're really trying to do here is it's a critical receptor for bone mineralization, but the bone hormone osteoclastin is very efficacious on bone maintenance, but activation of vitamin D receptor also activates the calcium channel, which when people take too much vitamin D supplements or hypercalcemic, that leads to bone pain and actually kidney toxicity, which can be fatal. So there were primary assays that were being used to find compounds that separate activation of these two targets. I'm going to skip that guy. We scanned a whole bunch of ligands, doing this 2-key analysis here. The one nice thing about showing this graph here is that these were 3,600 injections or 300,000% D values, and it's just basically the P-adjusted 2-key values here to show which ligands were more similar to others. So we didn't know what to do with that at first, but then we thought, why don't we do linear regression and compare this to a functional output assay? In this case, it was qPCR-based assay looking at TRPV6 expression. And lo and behold, there were peptides within the data set that showed a nice correlation between their protection and the functional output of TRPV6. And basically, you just walk up here where you have a compound that looks really good. So all these were potent on the bone hormone, but this guy basically does not activate TRPV6, but it also has essentially no stabilization of helix 3, whereas the natural hormone, which activates both, shows very strong perturbation. So that started a project which G is going to now walk through and show an example of VX1, and then in the last little bit cover cross-linking.
Sure.
My understanding is
Just the calibration is done?
No, you're labeling at whatever pH you want. So say your protein's happy or active at pH 7.5, we're using labeling at pH 7.5. So at our 10-second time point, we quench that with a low pH, low temperature buffer to stop any further in and out exchange. That's then fragmented into peptides and measured. The data is reflective of what labeled in at pH 7.5. Yeah, sorry.
Is there any other measuring stage going on other than that? And are there other ones that?
Yeah, so all the side chains are exchanging, but because of the time frame between proteolysis and trapping on the cartridge and the reverse phase, they fully back exchange. So under flow exchange conditions, they're not protected to back exchange. So they just back exchange out completely. So they're gone. And we do run 100% deuterium control, so that tells us that in fact has happened. So if there's 8 amide hydrogens, when we run the 100% sample, we should get 8 deuterons, and for the most part we do. We do lose deuterium across the platform, the time. So the longer retention peptides lose more deuterium. So that's why we use that 100% to correct the value back up. But the earlier eluting ones usually are close approximate 100%.
So what types of flexibility can you grow with HDX? Informational domain flexibility, local flexibility, what?
Yeah, it's all the, because we're digesting it and getting regions of protein. Now, more global, the EX1, you'll see an example here where that might be looking for more coordinated unfolding of a protein, like a hinge region of a protein. But within the subunits, you'll see the conformational flexibility.
Can you tease out mixture of conformations?
So if you have 2 distinct conformation populations, you're going to get the EX1 characteristics, which you'll see in a second. If there's subtle, we're seeing everything at once, so that is hard to tease out. But if you have, say, a molecule that stabilizes one conformation versus another, then maybe you get some insight.
In terms of probing interactions of a molecule, another molecule, is there a limited cutoff of K off that you can follow in terms of time? How slow?
Yeah, so we do it at equilibrium conditions, but so the affinity is usually would be the cutoff. We've done fragments so that we're high micromolar affinity, but with fragments we can add millimolar fragment to the sample. We just need to drive it to the bound state. So if it's only 50% bound, we're going to be 50% apo, 50% bound, and the signal's going to be really small. So we try to drive it to the bound state, so we typically want to be about 10x over the kb. Most experiments we do, there's no idea what the kb is, so we just add a lot. So now with protein-protein interactions, adding a lot of one protein over the other, one could be a limit by you can't get the protein at a higher concentration. But it also then complicates the experiment a little bit because you have so many peptides. Actually, what happens is not so much the interference in the mass spec, it's the digestion efficiency of the other protein goes down.
But the more you increase the concentration, you can also
you
have the problem with having self-assembly.
Yeah, I think the other things going on there. So, things that drive aggregation. You usually see that by loss of signal in the experiment. If the aggregate is precipitating or something. So, yes.
Do you ever do any studies with just lysate, or is the signal too poor?
Yeah, we haven't done that yet. So the purity question hasn't come up. Purity really isn't an issue for us at all. It's whether the target protein is at enough concentration that we get really good coverage. So if you can get your protein in a lysate at high enough concentration, we should be able to see it. We haven't done those experiments yet. There's some published papers on cell, direct cell. It's all been on red blood cells.
Okay.
Where protein is highly abundant and there's fewer proteins in the sample. And they've only really done like one time point. So it's like measuring immune protein overexpressed at one time point. Just showing it's different. So you can potentially do the experiment. It's just tough. Now we've done isolated complexes from cells and directly analyze those, not overexpressed like in E. coli or something. That was squid rhodopsin. That's a little cheating. The amount of rhodopsin in the squid eyeball is huge. But we've done other cases where it was quickly IP out of a HEC cell line overexpressed, looking at a complex.
Yes.
Can you, how reliable is to map the residue, the hydrogens that are participating in interaction between between a molecule and a drug?
Say that question.
How reliable is the method to map the hydrogens that have been exchanged, or in other words, to map the hydrogens that are participating in
Directly with the interaction with the drug?
Yes.
Yeah, that's tough. So because again, the effects can be just stabilizing the hydrogen bond network, and that's typically what we're measuring. I just showed the example with BDR that there was a general agreement of the regions correlated nicely with where hydrogen bonds are made, but again, it's within the peptide, so we don't know exactly which region is. The ETD experiment that I showed, I went through that really quick. That was plus or minus a ligand, and we knew based on the co-crystal structure that the ligand made a hydrogen bond in that particular amide, and that's where we sort of step up. So potentially with the sublocalization approaches and the Bayesian reduction to the single amide, you can find with enough experiments that there's a particular amino acid that's involved with interacting with the ligands. So, but that's not trivial.
Mhm.
And often we would confirm that by mutagenesis. So we had, I didn't show the example on PPAR where with a series of ligands, there was a difference in the enantiomers in one peptide, and it sublocalized to a phenylalanine. And when we mutated that, so one enantiomer was an agonist, the other one was an antagonist. When we mutated the phenylalanine, they were both agonists. So it was clear that they were clashing with that amino acid. So.
Okay. So, okay, I added some point to previous question, especially during my interview to give talk for faculty position. A lot of people asking, can you guys really do deuterium exchange on cells? But in my answer is, cells they don't like deuterium buffer. Like most mammalian cells, they can survive 40 minutes or 1 hour longer. And for bacteria, which is fine, they can survive in deuterium, which is like the red blood cells. So in our case, we can also try to look at overexpression of GPC on the membrane, cell membrane to capture the time frame of 30 minutes time frame to doing our experiment, since these cells should be alive during this deuterated buffer. And so, oops. Okay. Right. So I think one of the important questions of interest from our study is why we use deuterium exchange, because all the proteins, they're under their physiological conditions, they're not rigid bodies. And our deuterium exchange data can be totally complementary to X-ray crystallography and also cryo-EM studies. And also there's huge demand in the industrial application where deuterium exchange can directly map protein dynamics and protein ligand interactions, where they look at SAR studies with huge amount of compounds that derive from the lead compound in the drug discovery procedures, and during exchange can really push and speed up the drug discovery procedure. In this case, I'm going to talk about a little bit more about the collaboration between our lab and the Eli Lilly company, where we're trying to look at the safer vitamin D therapeutics targeting osteoporosis. So the vitamin D is a nuclear receptor can master regulation gene expression program regulating the calcium homeostasis. And the 125D3 is very strong agonist, can upregulate gene Big Lab, which is the bone hormone that is beneficial for the bone density. However, the overexpression, the dose-dependent use of this agonist can also trigger overexpression of HIVE-6, which is bad gene activation, since in the intestine is a calcium channel, which can increase the blood in the calcium concentration in the blood sample, which can cause the kidney stones. So we don't want this happen. So we collaborate with Lilly, and they provide very dissociated compound which can only activate the Big Lab gene activation, whereas to minimize the gene activation of HIVE-6. So we screen a lot of compounds, and finally they give us 3 compounds in direct comparison between the 125D3 and the compound 1, 2, and 3. Where in this case you can see there are very selective or dissociated profiles between the Big Lab and HIVE-6 gene activation programs in terms of the where's the mouse? So this is very potent in terms of EC15 values, and the maximum gene activation, maximum sting value is about 100 percentage. However, in this compound 3, gene activation is about 75%. However, the HIVE-6 we can be minimized to less than 20%. So this compound is really a dissociated compound that we're looking for. So we're using during exchange in combination with crystal structures to perturb what's the protein dynamic changes upon binding to these compounds. However, in our ligand binding domain crystal structures, we try to line out all these 3 compounds and do a silico docking. And what we found is all the structural orientation within the ligand binding pockets very, very similar. And however, we do know there are a bunch of different protein HDX perturbation going on between within this pocket. So we designed our experiments looking at the full-length vitamin D and oxal hydrodimer in presence of different drug molecules and DNA response elements, and also SRC1, the downstream interacting co-activated protein. So we can actually do a massive deuterating characterization looking at 26 differential intact protein complexes at the same time, and directly compare between each conformational states. So these are the conformational layouts of all the studied conformational status. So here you can see the ligand binding, DNA binding domain, and Henry region, and the ligand binding domain. We're looking at the protein ligand interaction in the ligand binding domain, and the protein DNA interaction in the DNA binding domain. So which is very straightforward and easy. And this is also perhaps the first story we try to incorporate cryo-EM studies, because several years ago, there's one group from French, they reported cryo-EM structures, vitamin D and oxal hydrodimer in presence of compound 125D3 and also the DNA response element DR3. And so this is cryo-EM density, and they fit in the density with the no ligand binding hydrodimer crystal structure, and also the known crystal structure of the DNA binding domain of the protein complex, and try to reconstitute a intact model. And so we asked this group for the primal version, and then we also try to improve the sequence homology sequence by asking our friend John Browning in Australia. So he gave us a primal structure which exactly mapped to our protein sequence that providing our that is done in our experiments. So this is the orange orientation and also the cryo-EM data of these vitamin D also hydrodimer protein complexes. And what we during my time in Griffin's lab, our laboratory is really pushed forward the direction to better visualization of our deuterium exchange data. So we have these protein HDX perturbation fields where you can, under each protein secondary sequences, there are different peptide fragmentations that Pat mentioned previously. And we can convert these into single amino acid consolidation field with the advantages of the smooth color gradient, so that we can to calculate what's the single amino acid deuterium exchange events, and we can map the this single amino acid consolidation field into the primal structure by our software. Now here's the demo of how our software can work out the protein. So first of all, we load our different directories into insert a primal structure that is known. So there are a couple of buttons you can click. So here's the button you can find and direct the place where a folder where you have the primal PDF files. And you can load the PDF files into our software HDX workbench. The whole procedure is very smooth. So after you can just load the whole deuterium exchange experiments, and the automation is directly made during the procedure. So this is the direct map of our deuterium exchange single amino acid consolidation field into primal structure. And so this is a consolidation field I talked about earlier. And the procedure is fully automatic, especially when you're doing large data analysis and can upload hundreds of primal structures at the same time, which is really convenient. So I did pretty much a similar procedure to vitamin D and oxide hydrodimer by this perturbation field and single amino acid consolidation field, and compare directly 25 different intact vitamin D conformational states in terms of in this vitamin D oxide hydrodimer. And one thing we can really in direct comparison between compounds, you can see in presence this compound, which is very dissociated compound, the threshold protection to deuterium is greatly reduced comparing to other organics in solutions. So, in industry characterizations, it's quicker to find out which compound is more potent in terms of protect to induce more HDX protection in the ligand binding domain. And another powerful how we can tell the story more interesting is that the deuterium exchange data can really tell about how the different domains about the intercommunications between different domains, like ligand binding domains and also the hinge domain, and also the DNA binding domain. In this case, in presence of the 125D3, which is the agonist, and if you add different DNA sequences, you can have two different phenomena. So if you have this DR3 response element, there's no crosstalk between the DNA binding domain and also the ligand binding domain. However, if you have another DR3 half set, so this is another sequence different DNA sequences. So you can see the crosstalk between the ligand binding domain and also the DNA binding domain. So which shows the protein kind of communication between each other in the same module. So this can be specifically studied by deuterium exchange. And I don't think the cryo-EM or the crystal structures can reveal such dynamic in solution or in terms of paper writing or story presentation. And to bring up the EX1 kinetics, where Pat mentioned earlier, so you have bimodal distributions where you can further divide into two different MS envelopes, where in terms of EX2 kinetics, so that you can only observe one single MS envelope along different HDX time points. And here's the characterization of the EX1 kinetics by the software HDX Express 2. And the location of EX1 of this peptide is located around this hinge region, where it plays a very critical role in determining the structural orientation of this ligand binding domain to the DNA binding domain. So where you can see the crosstalk between these two modules. So I'm going to some detailed data analysis of how we analyze deuterium exchange data. This one Pat just mentioned. So the intrinsic exchange rate is much faster than the protein refolding rate. The EX1 kinetics will take place. And on the other hand, if the protein refolding rate is much faster than the intrinsic exchange rate, we will only observe EX2 kinetics. So here is how we analyze the EX1 and EX2 kinetics, where you can convert the protein spectrum, the deuterium exchange spectrum into the digital numbers. So in the first column, which is the deuterated sample, so you have the M of Z values, and the second column you will have the intensity files, and then you can the HDX Express 2 can help you analyze the intensity and the map, try to fit into calculate the centroid of the different isotope distribution patterns. So I will take that. Oops. So here I have EX1 and EX2. It doesn't show up. Yeah, it can show up in the screen, but not the. Maybe we need to exit. There we go. So here's the direct data analysis of EX2 kinetics. You can plot out the percentage change of deuterium incorporations and look at different protein states. And so here's the undeuterate spectrum, where you can see most. And you can calculate, the software can automatically calculate the centroid here and give you each figure like the width, the peak width, and also the centroid, M of Z values. And if you do exchange data to incorporate more deuteriums into the spectrum, and to also give you another calculated centroid, so you can further use this centroid to plug out this deuterium uptake level, deuterium uptake HDX uptake plugs. Oops. Well, this is the EX1 kinetics, where I want to show the so you can use EX2 phenomena to fit the EX1 pattern because this peak width become wider, much wider, and there are 2 MS envelopes, which is very hard to fit by the regular EX2 fitting. So in this case, you need to play around several features in the software called binomial fitting. And after this fitting, you need to input the sequence, and also the charge state, how many amide counts within the protein's peptide sequence. And then you can fit the bimodal distribution and deconvolute the whole MS envelope by the first MS, so for the first MS envelope and the second MS envelope. And in the rear, this is EX2. So this is EX1. So in a rare situation, so the undetected isotope distribution should look the same. And however, in terms of bimodal distribution, you have MS1, the first envelope and second envelope. And you can, so when time goes by, you will see the first envelope become smaller and the second envelope become larger. And finally, you can have a summary of this plot, which we call it the bubble plot. So the red bubble dot represents the higher MS envelope, and the size of this the bubble represents the ratio of this of the second MS envelope, and the blue dot represents the first MS envelope. And this is the deuterium uptake calculations. This bubble dot can really help you to calculate the convert from the first MS envelope to the second MS envelope, so that you can calculate the structural change directly from this. So this is next one. Sorry, this computer doesn't work. So there's only one computer down. In the third computer working. If you guys interested, I can show you after this presentation, just doing real examples. So another one, let's try, we're doing a cross-linking MS. So we recently published a paper looking at different compound to modulate HIVE mobility by chemical cross-linking MS. Since our laboratory is the nuclear receptor laboratory, so we have different pharmacology compound from superagonist, full agonist, and partial agonist to partial inverse agonist and full inverse agonist. And the structural characterization of this HIVE 2 key part is very critical because this is ligand-dependent activation of this enzyme, and the structural orientation, the movement or dynamics of this HIVE is very important for enzyme activation. However, in our deuterium exchange experiments, in presence agonists, we can see protection in this HIVE and partially protection in presence partial agonists. However, in presence antagonists or inverse agonists, we can't obtain any information in terms of HDX protection. around this region. So what we think is whether we can use chemical cross-linking to cross-link the HIVE 12 to any other nearby or distal lysine, so that we can monitor the peak intensity of particular conformer of this HIVE 12 in order to calculate or to see whether there's more population of HIVE 12 conformers around different lysine cross-linkers. And we use the BS3 chemical cross-linker, which is commercially available, and also DDS3. So what BS3 is water soluble, and the reaction is the N-terminal amino group of this lysine can interact with the NS3 ester of this BS3 cross-linker, so you can form. And also the space of this, the length of this cross-linker is around 11 Armstrong, where the two lysines, the lysine has 6 Armstrong space arm, so the total length of one particular cross-linker reaction is around 25 Armstrongs. And we take all the protein and present different compounds, incubate half an hour in room temperature, and we add the 15 molar excess of this BS3 cross-linker just in a eventual solution, and waiting for an hour to reach to the equidistant conformational states. Then we quench the reaction by adding the trace buffer for another half an hour, and we can run the cross-linking samples by at the SDS page, where you can see all the samples become very merged and become very larger band. So this is the control protein sample, which is the band is very clear comparing to other smear bands. And we use the trypsin in solution digestion. For some proteins, we need additionally add up another second protease called chymotrypsin in order to induce more cleavage in fence to get a smaller MS peptides fragmentation. And we use LCMS, MS, just a standard proteomics workflow to characterize the MS files. And we use the software called P-Link, which is user-friendly nowadays and also free on the website. You can request from this group, and we can just use P-Link to characterize and extract the peak information for the cross-link peptides, and also calculate the intensity of these different cross-link peptides. And so all the layout of the lysine residues across PPAR 3D structures. And what is interesting is lysine on the HIVE 12 region. So this lysine 417.4, the position of this lysine can be, so we select another 5-6 nearby or distant distal lysines, for instance, K301 and also 285 and 457. So for distance from this lysine here to this 275 is larger, is around 35 Armstrongs distance. So it can be reached by cross-linking reaction if this helix is not labile in solution. If this helix is fixed in solution, the cross-linking reactions cannot take place. So we want to manipulate these dynamics of this helix 12 in solution, and looking at the protein ligand interaction, how protein ligand the compound can affect the dynamics of the helix 12. And we use P-Link 2 to finally identify all the possible cross-link reactions. And here's the summary. Since this in the apo state or in a non-organic bound state, this helix 12 is swimming around helix 3 region. However, in presence of agonist, it adopts agonist position where the helix 12 is docking against helix 11. However, in presence antagonist or further you add some co-repressor proteins, this HIVE 12 is swinging far away to the region near this region, so which is drastically different from this region. So the cross-link intensities between HIVE 12 to lysine here and is dramatically increased, which you can see the cross-link reaction here. So in these terms, we can see and calculate and study the protein HIVE 12 dynamics in solution by quantitative BS3 cross-linking MS. And this is very recent review articles, 2 review articles. I'm not going to illustrate more. And basically, you have protein complex. You can do in vitro cross-linking reactions, and you can do trypsin digest and do proteomic characterization, and you can have cross-link map between the secondary lineup of different landscape of protein or protein complexes. And you can do, finally, you can deconvolute the cross-linking data into a structural 3D model. We can also pull down the target interest and also do cellular proteome cross-linking reactions, and also in vitro protein complex protein big interaction, and to characterize similar workflow by proteomics, and there's statistical data analysis tools like force discovery, characterization, also visualization and modeling in the final step. And here I just want to list all the available different commercially available cross-linkers. There are DSSO, which is commercially available from the thermal, and this is MS cleavable cross-linker. And there are other cross-linkers that are specifically designed for attaching biotin tag, so they can purify the cross-link peptides from very, very large and complicated sample pools. And this is peeling to the software and paper they publish. Because the other softwares are not free, and this is really free. We can just ask for the license and key. They can ship you, and you can download on the website. If you want to buy Proteome Discover, which takes 10,000 US dollars, which is quite expensive. And here's just another demo for how to use the peeling tool. So they have a newer version called peeling tool to identify cross-link peptides derived from the APO regi, so which is a huge enzyme, and in the APO state forms auto-inhibited conformation between two domains. So this is secondary structure and layout. So this is CARS domain and also the HIVE domain and CTD domain of this regi protein. And previously, in a CAR2 module and the HAL2I module, there's this very strong static protein-protein interaction in the APO state, which locked the CARS domain into the HAL2I domain module here. So this is protein-protein interaction. So we want to use the cross-linking to capture this auto-inhibited conformation for regi. And there's no crystal structure available for this protein-protein interaction. So we use a purified regi molecule just in the APO state, and we add similarly as people are adding 50 molar excess of this BS3, including 1 hour, and a quanchit by cheese buffer, and add trypsin and do standard LC-MS MS calculations where we can get the these LC-MS MS data, and then we throwing into peeling tool for data analysis. And there are a couple of additional new features of the peeling tool, which is really user-friendly nowadays, and the speed, the data analysis speed is really fast nowadays, which is really simple. You just add the MS raw files for your protein, and you can manipulate these data parameters, and you can choose a couple of different commercially available cross-linker for you to choose from. So here I just choose BS3 cross-linker, and it's a database of your protein, so you need to find a faster sequence of your protein for interest. And here's enzyme, I use trypsin, and can up to 3 mis cleavage events for these protease reactions. And for any peptide masses, I'm looking for 600 to 6,000 around these Dalton masses. And the peptide length is between any peptide length between 6 and 60 will be the target for the software searching. Since we analyze the bacterial protein, so there's no more further post-translational modification of this protein. You can also have a filter the tolerance, false discovery, and also the MS accuracy of 10 ppm for the data confidence throughout identification. And here is summary, and basically just hit run, and this one will process around 20 minutes. So you'll have the data. Here's the data, statistical data for all the peptides and also cross-linking peptides identified by the software, which is really accurate because the precursor mass errors around is less than most dots are captured within the 2 ppms, and also the false discovery is less than 1% here. And they will generate some Excel files for you to further detail the data analysis, where you can manually analyze the data. They can also have a visualization function where you can see, for in this case, I want to see the this CAR2 module and the HER2i protein autoinhibition between lysine 80 and the lysine 528. So here's the lysine 80, and this is lysine 528, where you can capture by this particular peeling 2 program. And there are other different cross-links between different lysines and different lysines between these 2 sub-modules I didn't show here. But in this protein, I identify about more than 80 bona fide cross-link reactions that can be validated from the structure. And for the summary, and also for the HDX section, so the deuterium exchange can provide direct insights into conformational ensemble, mobility, flexibility of the protein, and it's good for protein enzyme activation studies for any enzymes, and it's complementary to the cryo-EM and also crystal geography. Here's the last slide.